This is deoxyribonucleic acid, or DNA. It is wound in anti-parallel strands. Notice how the strand contains a 5' end and a 3' end, as the strands run in opposite directions. This is a nucleoside triphosphate, the building block of DNA. These are the nitrogenous bases, which are split into two groups. Pyrimidine is an aromatic heterocyclic organic compound similar to pyridine. This group contains cytosine, uracil, and thymine. A purine is a heterocyclic aromatic organic compound that consists of a pyrimidine ring fused into a imidazole ring. This group contains adenine and guanine. This is a replication bubble that contains a replication fork. It is where DNA replication is mechanized. This is the semi-conservative model for DNA replication. Notice how the parental DNA is used as a template for the daughter DNA. Topoisomerase breaks, swivels, and rejoins the parental DNA ahead of the replication fork, relieving the strain caused by unwinding. Helicase unwinds and separates the parental DNA strands, as single-strand binding proteins stabilize the unwound parental strands. Primase synthesizes RNA primers using the parental DNA as a template.
RNA primer is made. DNA polymerase III starts to synthesize the leading strand. The leading strand is elongated continuously in the 5' to 3' direction as the fork progresses.
Primase joins RNA nucleotides into a primer. DNA polymerase III adds DNA nucleotides to the primer, forming Okazaki fragment 1. After reaching the next RNA primer to the right, DNA polymerase III detaches. After fragment 2 is primed, DNA polymerase III adds DNA nucleotides until it reaches the fragment 1 primer and detaches. DNA polymerase I replaces the RNA with DNA, adding to the 3' end of fragment 2. DNA ligase forms a bond between the newest DNA and the DNA of fragment 1. The lagging strand in this region is now complete. A thymine dimer distorts the DNA molecule. A nuclease enzyme cuts the damaged DNA strand at 2 points and the damaged section is removed. Repair synthesis by a DNA polymerase fills in the missing nucleotides. DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. This DNA repair system is called the nucleotide excision repair. The adjacent hydroxyl groups on DNA fragments help indicate where to replace RNA with DNA. However, at the end of the chromosome, there is no hydroxyl group to indicate this. Because of this inability, DNA would replicate shorter each time it is copied. To solve this end replication problem, telomerase kicks in. The ends of DNA contain a G-rich series of repeats called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the strand in a 5' to 3' direction and adds additional repeats as it moves along the strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. This allows for the replication of the entire DNA strand. Fragment 2 is primed, the DNA polymerase